Okay, so today, so it was kind of a rough one last time with all the updating, and what what I forgot to sort of mention last time, just as an overall kind of overall picture of what's going on, is this idea of efference copy. So this is a very old idea. This, it's a 100-year-old idea. Efference copy. So what is efference means? Some information that's leaving the nervous system and sort of doing something. And copy presumably is like, copying what you're planning to do. And so this updating thing that we were talking about with the double step saccade is like a prime example of this efference copy. So the idea is you've got, the superficial superior colliculus and deep superior colliculus, and those project down the deep that projects down to the deep, and then this goes to H and V, horizontal and vertical, and then that goes to the oculomotor neurons. And the oculomotor neurons then go out and actually sort of hit the muscle. That's supposed to be a muscle. So what is efference copy? Efference copy is you do a whole bunch of computations, figure out where the targets are, maybe they went off, keep them on, blah, 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 move them around. And then you feed back a signal. So this is the efference copy part. You feed back a signal of what you were intending to do. So you go and get the final answer. So this is the final answer. This is what you're actually going to do. After you've done all your computation, you come up with the final answer. The final answer is we're going to move the eyes to this position. In the process of moving the eyes to that position, you peel off a copy of that signal and feed it back into the system, and that essentially tells the rest of the system what you just did. Now you could actually try to figure out what you just did by sort of like doing some sensory processing and finding out where your eyes went to, or you could try to measure with muscle spindles where your eyes went to. But this procedure of just essentially siphoning off the final answer and feeding it back into the system is a critical sort of concept, and that's called efference copy. And there's a lot of examples of it in the motor system. And so what we want to do today, getting the eye position to come back into the colliculus and move the targets around, that's a prime example of efference copy in action. And so that information coming from the oculomotor neurons is the thing that comes back into the colliculus and effectively sort of moves the hallucinated targets around so that they're in the right place. So this was a too simple diagram. So today we're going to actually look at, I don't expect you to remember all this, but we're going to look at, sort of back out and just look at all the parts that are involved. So when you do scientific research, you typically use a reductionist approach where you go someplace like into the deep colliculus and record some neurons in the deep colliculus, and you manipulate the conditions, and then you try to sort of figure out what's going on in the deep colliculus. But the problem is there's about 50 parts. Question? This guy? Oh yeah, that's supposed to be an eye muscle right there. So there's the eye, and this is an eye muscle. Sorry. Yeah. So I mean that's the way you get science done. You focus on a particular, say the deep colliculus, and do some recording there. But the problem is, the deep colliculus is hooked up to about 50. Literally, it's hooked up to about 50 things, and so let's just sort of just put them all out there just for the hell of it, just to get some idea of like what the whole system actually looks like, and how the efference copy kind of just invades the whole system and goes all the way up into the cortex, and then we'll talk in the second half about all this, how this information is used in the cortex, and when you actually have to combine information from more than one modality. So if we start off, last year I scrunched it a little bit too much, so let me just I'll go to here. So basically we start off with the eye. So here's the eye. There's the retina, and then here's one of the eye muscles. When you scan somebody, you can see the eye muscles. They're big, long things that kind of extend into your eye socket behind your eye. And so we diagrammed. If we look at the retinal input, we diagrammed several pathways, sort of parallel pathways. So there was write it out because it's superior and superficial are kind of overlapping. So superficial, superior colic. And we said that those pathways, you have some input coming in from the retina, and it goes to the dorsolateral geniculate, and it goes to the superficial colliculus, and then the dorsolateral geniculate projects to V1, and that goes to V2 and MT and MST, a lot of cortical areas. And we said that sort of V1 and all those cortical areas are kind of similar in function to the superficial colliculus. So these guys have a similar function. So they represent where stuff is, where the objects are. And then they have output to motor structures. So I'm going to separate off the intermediate superior colliculus, because in the superior colliculus you kind of have like a whole gradation of really sensory things on the top, and then things in the middle that are kind of more like salients that don't necessarily have to have something there, but it's still sort of a sensory-like signal. And then you have the motor signals in the deep colliculus. So intermediate superior colliculus is kind of in between that. And then here's the deep superior colliculus. And the deep superior colliculus is really motor. It only fires right before an eye movement. And so those are connected up to each other. So superficial colliculus sort of goes down to intermediate and to deep. And then that thing goes down to the, let's see, where am I putting that? Yeah, okay, so I'll put that over here. So then we have the deep colliculus that actually goes to the pattern generators that generate the bursts that are, let's spread this out just a tiny bit. So the rostral interstitial nucleus, with no N, of the medial longitudinal fasciculus. That was V. You don't have to remember that one. So that guy is vertical. And then we had the paramedian pontine reticular formation. That was the horizontal. So those guys are the pattern generators. So these are saccade burst. It just makes the burst that sort of jerks the eye to the new saccade burst pattern generators. So those guys are just making the burst of the eye, and the colliculus the deep colliculus projects to both of them. And then we said that the thing that's like the deep colliculus up in the cortex is the frontal eye fields. So here's the, let me give myself some space here, yeah. So here's the frontal eye fields. And the frontal eye fields are kind of functionally similar to similar function to the deep colliculus. And the frontal eye fields have their own separate access to these low-level output structures. So what does the frontal eye field get its input from? Well, there's a lot of these areas like MT and MST, project up to the frontal eye fields, similar to this projection. And there's, several stages, so there's kind of an intermediate colliculus-like thing going on in some of these higher visual areas. So that was basically the saccade circuit. But remember, I separately drew the other circuits because there's basically an input to the pretectal nuclei and the terminal nuclei. There's 3 of those. So like say 2 pretectal, three of these guys. And those guys get their own separate retinal projection. So there's a separate retinal projection that goes down to these guys. And these are the ones that are involved in the optokinetic nystagmus. And how do they sort of do the optokinetic nystagmus? They project to the vestibular nuclei. And we said that the vestibular nuclei, they're called that because they get input from the semicircular canals, but they're really sort of more general than that. They're sort of like the general purpose, multisensory, combine information and sort of stop you from falling down nuclei. So there were 4 of these guys, 4 of those guys. And so the pretectal nuclei project to them, and then the vestibular nuclei project to the oculomotor nuclei. There were 12 of those, 6 for each eye. So there's the oculomotor nuclei. And then the oculomotor nuclei go out, back out to the eye muscle. So goes to the eye muscle to actually move the eye. And then we also have the vestibular, there we go, vestibular input. So here's the vestibular canals. Let's just draw those. A vertical one. So there's a ganglion cell hooked up to a hair cell. And that guy comes in and projects to the vestibular nuclei. So that goes in. That's where the vestibular nuclei get their input from. And so these guys were kind of doing the OKN. And then the vestibulo-ocular reflex, which was to sort of cancel head rotations. This was like cancel movements of the whole world, OKN. But the vestibulo-ocular reflex is also done by the vestibular nuclei. And then we also had the cerebellum in there. So here's put it down a little lower. So we've got the cerebellum in there, kind of smaller than it actually is. And the cerebellum, projects back and forth to the vestibular nuclei, and it projects back and forth to the terminal nuclei, and that's how the information on smooth pursuit gets in. So we have the pontine nuclei in this little hole right here. And information comes down, from MST and MT down to the pontine nuclei, and then that gets to the cerebellum, and then from the cerebellum it can get into the vestibular nuclei and cancel optokinetic nystagmus, or go to the pretectal nuclei and tell them to stop doing your optokinetic nystagmus, because whenever you do the pursuit, so this is, we'll call these guys, pursuit. This is like the pursuit pathway. When you're following a small object, you've got to cancel this follow the whole world business, and so that's how that gets in there. But there's more. So we've got. I guess I have to make a small hole in my diagram right here. Apologize for this. Okay, so this is the oculomotor nuclei going to the eye muscle, and here's a visual input going to the pretectal. That's just enough space. So the striatum is involved in eye movements. About a third of the striatum. Now you think of the striatum like the basal ganglia, like it's drug, sex, and rock and roll, and reward, and things like that. But there's a big part of it that's involved in sort of gating sequences of movements, and that. So if you look in the basal ganglia, roughly a third of it is involved in eye movements and head movements. So it's a major thing. So what is, didn't make enough space. So it turns out the entire cortex projects to the basal ganglia. So, okay, it's wrong color. So here's the oculomotor going to the, and here's the retinal coming into here. Okay. So here's the caudate and putamen. Those are, we haven't talked about those yet, but those are parts of the basal ganglia. And the caudate and putamen essentially receive input from the entire cortex, literally. The entire all of the cortex goes to the caudate and putamen. There's literally no part of it that doesn't go there. And then that projects to the globus pallidus. Yeah, I'll leave that out. That also, in addition to projecting to the globus pallidus, it projects to the substantia nigra. Substantia nigra, the black substance. And so that, again, you associate the substantia nigra with reward because there's dopamine there, and antipsychotic drugs mess around with the dopaminergic input to different parts of the brain. But the substantia nigra, a bigger part of the substantia nigra, is a GABAergic part of the substantia nigra, and that's actually heavily involved in sort of gating eye movements. And it gets its input from a number of places, but the caudate and putamen is one big place. And like I said, the caudate and putamen essentially get input from the entire cortex. So let's just put the entire cortex up here. Not just, V2, but the entire cortex. So there's the whole cortex. And so that projects, so literally the entire cortex goes down to the caudate and putamen, different parts of it. And that projects to the substantia nigra, which has two parts. There's the dopaminergic part, and then there's a GABAergic part that's an inhibitory part. And that projects, that's substantia nigra, projects to the intermediate and deep colliculus. And it turns out this is very important for sequencing what you're doing. So if an example is something like the substantia nigra and the caudate putamen, the kind of thing that they do. If you're tracking something around, like like there's a monkey, moving around, you're tracking that guy, like where is he? I'm tracking him. And then you decide, oh, I don't care about him anymore. I'm going to start tracking somebody else, somebody else that's crossing there. Let's go over and start tracking this guy, making a bunch of movements. You'll find that the substantia nigra, the GABAergic part, is strongly activated when you transition between those two sort of movement sequences. So it's kind of it's very clearly involved in kind of segmenting behavior, hierarchically segmenting behavior, so that you actually say like, oh, okay, we've been tracking this guy, a bunch of eye movements back and forth, but now we're going to break off tracking that guy and start tracking this guy who might be in the same place as the other one, but there's kind of a cut. Now we're tracking a new target. So that's involved in this, in this whole system here. So, so what do you want to call this? I'll talk about this later, but with, the cauda imputamen and the substantia nigra are you can sort of think of them as sort of hierarchical motor control processes. You have like little walking steps. Those are a little pattern, but then you're going to make a turn. Like then there's a cut in your behavior, and you make a turn, and then you make a bunch of little walking steps. Or you have like a really big segment, like I'm planning to get Thai food tonight, and that could take like a couple hours, And so you've got that's a goal that's kind of running like, get Thai food. And so then you slowly play out all these different things, like drive to the Thai restaurant, as sequences of behavior. So that's the kind of thing that the cauda imputamen does. Okay, but what are we going to talk about today? Well, we'll talk a little bit about the colliculus when we talk about these multisensory interactions, but we'll also talk about some other areas in the cortex. And those other areas in the cortex are, one of them is called VIP, because it's the ventral intraparietal area. I'll make a little diagram of where it is. So it's in the intraparietal sulcus in a monkey, and it's in the bottom of the sulcus. That's why it's ventral. So VIP, I'll talk about. It's a multisensory area. We'll talk about some things that are going on in that multisensory area. And then there's another area called LIP, for lateral intraparietal area. And we'll talk about how you get some predictive updating there that's coming from the colliculus. So ventral intraparietal area, it's getting, it's a visual area, so MST projects to it, MT projects to it, but it also gets input from somatosensory areas. So here's area 5, that's a somatosensory area, or area 7B, that's another somatosensory area. So those guys have to do with actual touch, and those guys also project to the ventral intraparietal area. So these guys are somato. Somatosense. So those guys provide multisensory input. Now, one of the things that you might immediately realize is if you've got your face input and then your eyes are moving around, it could get out of alignment. And that's one of the things we're going to talk about, because it's combining information from somatosensory and then visual information coming in. Okay, so since we're talking about VIP, where does it get some of its information from? Well, it gets information from the frontal eye fields. That's kind of a backwards projection, because the frontal eye fields in the frontal cortex, and they're going sort of backwards to ventral intraparietal. What about LIP? Well, LIP gets some input from there as well, and of course, LIP gets some input from MT and MST as well, some visual input, and LIP projects to VIP too. So that's a reasonable inventory of the areas that have coordinated activity in them. throughout all these areas for any eye movement, every single eye movement. So a lot of times you sort of think of the brain, you've got something doing eye movements down there, and then you have the cortex sort of studying truth and beauty up here. But in reality, what it is you've got the eye movement machinery down here moving eyes, and that information is instantly propagating up every time you move your eyes up into the cortex, and basically telling the cortex what the world is going to look like after you move your eyes. And that it's thoroughgoing. It covers all different areas in the cortex, immediately covers all different areas in the cortex. So there's kind of like a coordinated system. Now the problem is, how could you possibly understand this? Nobody has actually recorded from all these things at the same time to see how it really works. And so to some extent, I'm just kind of making up stories. People have recorded the frontal eye fields, and we'll have the animal move its eye around, but you don't really know, what was the substantia nigra doing when you were doing that? You didn't record there at the same time. Or, what was the intermediate colliculus doing? I left out one thing. So there's another nucleus here in the thalamus. No, it's a little too high for the thalamus. We'll put it down a little bit lower. So the mediodorsal thalamus. So that's actually thalamus. Let's put a label here. Thalamus. And the deep colliculus projects to that. So the deep colliculus projects to that. And it turns out that's actually the way that the eye position information gets up to the cortex. So medial dorsal thalamus projects to the frontal eye fields. So we're going to see in VIP, we'll talk about VIP and LIP, that you have eye position information coming and helping line up the maps of your face versus maps of the visual world as your eyes move around. How does that information get into VIP? Surprisingly, it comes through the deep colliculus to the medial dorsal thalamus. Let's write it out. Medial dorsal thalamus. And then that projects up to the frontal eye fields, to frontal cortex, and then it goes backwards to VIP. So that's how the actual eye position information gets into the cortex, to these posterior cortical areas. People didn't think it was that way. They just assumed that it was probably doing something, in the cortex. It wasn't, but it's being directly driven from the deep superior colliculus. Now, I started off saying efferent's copy, but I didn't mention the efferent's copy. Where there's still some space down here. So where does the efferent's copy come from? Well, if you look in the oculomotor nuclei, what you find, you find some large neurons. So this is oculomotor. Drawn again, oculomotor. So you find some large neurons in there, and those guys are sort of going out and hitting the muscle. Those are the large motor neurons. But then you find some other neurons, these smaller neurons that are just sitting in there, and they don't actually project out. And those guys are called the, so in the case of the abducens nucleus, this guy is an abducens, So that guy moving, that abducens means abduct, which means sort of move away, like move out, move laterally. So that's the abducens motor neuron. It's just one of the nuclei. And then that little guy is called, it's actually not separated, but it's just sort of sitting in there. It's called the accessory. Accessory abducens nucleus. And that guy just samples the signal, the final answer. Like what signal got sent to the abducens nucleus to move the eye. And it feeds it back into the system. And so where does it, so it's basically something, you've got the the big motor neurons here. That's the big motor neuron in the oculomotor nucleus, and then here's this little accessory abducens nucleus embedded within the main abducens nucleus, and that thing projects out to a ridiculously named structure called prepositus hypoglossii. Hypoglossii. So what is that thing? So what does that mean? This just means something that's in front of the hypoglossal nucleus. I didn't make the name up. Okay, so that one is mainly for the horizontal position, but there's a vertical equivalent, which doesn't even have a name to my knowledge. There's a vertical equivalent that's basically sampling the vertical eye position that went out, and so those guys get input from those accessory abducens neurons that are sampling like what was the final answer. So that goes back out here, and what this has in it, this has horizontal eye position. It has to somehow get rid of the burst. So remember, like the neurons, they're firing continuously, and then there's a sharp burst, and then they'll end up at a new firing rate, something like that. So they somehow have to cancel out the burst and just find out this, like what is the eye position. So there's some processing going on. We don't know exactly how that works, but somehow these guys, if you record from them, they just tell you what the eye position is, the current eye position is. And so then that information gets fed back to the midbrain tegmentum, which just means the floor of the midbrain. And sometimes this is called profundus, the deep mesencephaly. The deep midbrain. So that's where these guys go. And there's some other inputs that come from the vertical equivalent, and then that gets fed back into the deep colliculus. So it's supposed to be green. There we go. And that's basically how the double-step saccade works. So that's the way that the information about where your eyes went to is fed back into the colliculus in order to update all those targets, because it has to somehow like sort of rotate the little planetarium around in the colliculus to take account of what eye movement you made. But not only does it do that, it then goes up to the medial dorsal thalamus, and then the frontal eye field, and then VIP, and you see it does stuff all over the cortex. And this is basically the explanation of why the world doesn't jerk around when you look around. All your cortical areas are being touched by the deep colliculus. Okay, so this is eye position is coming out of this. Okay, so what are some of the things that are done? So I'll talk about three in the second half here. So what are some of the three kinds of particularly multisensory interactions that this information can be used for up in higher cortical areas? So the first one, how should I do this? I'll do it side by side. Okay. So the first one is a VIP vis som align. Okay, so if we look up in VIP, so VIP is a high-level area in parietal cortex, and it's got a representation of touch, not just the face, it actually goes down the body, down all the way down to the toes. And then it has a representation of touch, but it also has a representation of visual objects. It's especially responsive to visual objects that are close to you, like, within like 6 inches or a foot. But one of the problems, if you consider that area, is that it's got visual input, and then it's got somatosensory input from the face, but the eyes can move around. And so if you want to correlate information about a visual object, and then, like a leaf that's coming, maybe just touching your forehead, you can see it, and then you can also feel it. But if you want to correlate those two signals, you somehow have to take into consideration where your eyes are pointed to, because, and this is particularly important if somebody's like, say, throwing something at your head, or something is like, it's important for walking through a door frame so you don't bang your head on the door frame. So what you see is, your eyes are going various places as you walk through the door, and then the door target comes in visual coordinates. But what you need to do is make sure I don't bang my head into the door frame. And the door frame is going to be in different positions in the visual map, depending on where your eye movement takes your eye movement to. But what you want to know is, where is the door frame relative to my head? And so that's why people have, said that, we have a head-centered coordinate system. So you can think of it as really somatosensory centered. So it's basically sort of fixing up the visual information back into somatosensory coordinates so that I can avoid banging my head. Or if I want to bite something, it doesn't matter where my eyes are. It's like if I take a spoon and bring it up to my mouth, I might be looking slightly to the left or slightly to the right, in which case the spoon is in, say, left V1 versus right V1, but I don't care about that. What I really want to know is, like, where is the spoon relative to my mouth? I don't care where it is in my visual field. And so we somehow have to convert our visual signals into signals that are visual signals relative to the head. And that's what VIP does. And so I always draw my little Homer Simpson monkey. So here's a small nose, large mouth, eye there. And so you have that monkey that's looking out at the out at a screen. So here's a screen that the animal is looking out at, and say the animal is just looking straight ahead. Now, if you go and record in VIP, so where is VIP? Well, if we look at let's make a quick drawing of a macaque monkey here. That's the central sulcus. That's the intraparietal sulcus, and then here's the frontal sulci and the STS and the inferior occipital lunate. So this is the intraparietal sulc. So where is VIP? It's kind of like down in the intraparietal sulcus, right down there. So that's VIP. It's in the very bottom of the sulcus. In humans, it's a little bit more medial. We found it in humans by air puffing people's face to try to figure out where this area is, and it's a little more medial in humans, but that's where it is in a macaque monkey. So if we record from that area, we find out it's got visual input and face input, or upper body input, like shoulder input or sort of stomach input. And so if we record, you'll find two different receptive fields. So say you find a receptive field that's on the chin of the animal. So that's basically the somatoreceptive field. So you just touch with a little brush, and you can see, it responds like when you just stimulate this part of the chin. If you then go and look at where the visual receptive field is, when the animal's looking straight ahead, you'll find the visual receptive field. So this is visual receptive field. you'll find out it's aligned. And what do we mean by aligned? Well, it's sort of in the same place in the world. So that's, aligned. So the visual receptive field is aligned with the somatosensory receptive field. And so if you find a receptive field on the forehead, it will a touch receptive field on the forehead, it will correspond to an upper field visual. If you find a receptive field on the nose, it will correspond to a central receptive field. But you can see what the problem is. The problem is, what if the animal looks somewhere else? And so, which you do all the time, is constantly looking other places. And so now you just have the animal just look, small nose, large Homer Simpson mouth. You just have the animal just look up. So exactly the same setup, but now you just have the animal do a task where it has to look up. So what's going to happen? So there's several possibilities. So if the visual receptive field the visual receptive field, typically, if you think of it in the case of the lateral geniculate, it's kind of attached to the retina. So wherever the retina goes, it takes the receptive field with it because it's coming from a particular part of the retina. And if that was the case, where would the receptive field go? Well, it should go like about that far down. So it should be like about there if the receptive field was attached to the retina. But it turns out what happens is the somatosensory receptive field doesn't move. It's still on the chin. And the visual receptive field actually gets moved. So it actually gets moved to over here. So visual RF moved to align with somato. So basically what happens is it actually sort of adjusts the visual input so that it will come in and actually line up with the somatosensory input. Now, it could have done it the other way. It could have said, oh, you're looking up, the receptive field has gone up, and so I'm going to move the somatosensory receptive field onto the nose, something like that. But it doesn't do that. It basically just moves this guy over here so that it lines up with the somatosensory receptive field. And why is that? Well, what is the goal of VIP? The goal of VIP is to not get your head hit by a visual object, or it's to bite something, regardless of where your eyes happen to be. So there's an example. So what does that? So, eye position does this. And how does that eye position information get there? Well, and remember, it's just like it's happening. You have to sort of think of the whole system. The whole thing is coordinated, and all these different maps are being coordinated. So every time you move your eyes, some information comes from the oculomotor nuclei, gets into the prepositus. It sort of moves stuff in the deep colliculus. The deep colliculus projects to medial dorsal nucleus. That goes to the frontal eye fields, and that goes to VIP. So that's where the eye position information is getting in to VIP to shift the receptive field, the visual receptive field around. So it's actively sort of shifting where a stimulus in some part of the retina is going to. So in other words, like instead of this, a particular part of the retina going to this neuron, it's making a different part of the retina go to this neuron. So it's an active process that happens every single time you move your eye. So that was a case where we moved the visual map to line it up with the somatosensory map, because the goal of this area is to sort of like, it's a somatosensory goal, trying to not bop your head and if somebody throws something at you, it doesn't matter where your eyes are, you need to sort of like figure out where is it going to hit on your head. Or if you're trying to bite something, where is it relative to my head? I don't really care where it is relative to my retina. So that was the first one. So second one, let's go down into the superior colliculus and look at B. So let's look at auditory inputs to intermediate superior colliculus, intermediate SC. Okay, so we've got a similar kind of multisensory problem going on with auditory localization and the eyes. Because, so what are you using this for? You're using this for something that you do all the time. Something makes a noise, let's look at it. So you can do this effortlessly. It doesn't have to have a visual representation. A noise could come out of something like the refrigerator. It doesn't make any visual signal. And you want to look, let's look at the refrigerator. What was that noise come from? So in order to do that, you have to take account of where your eyes are looking, because your eyes can move independently of your ears. And so this was a side view. So now we're going to do a top view. So now we'll do a top view. So we set up an experiment where we have an animal. So here's an animal. There's its ears. Its head is fixed. And it's looking out at a screen. And we'll have make it a slightly smaller screen. So we'll have two conditions. So in the first condition, the animal's looking straight ahead, and a target comes on. And there's two kinds of targets that could come on. You could either, put a light on, or this is supposed to be a little loudspeaker. You can put a loudspeaker on there. And so in the loudspeaker case, we put just a sound there, and then the task of the animal is look at the sound. Look at where the sound was coming from with no visual signal, just in the dark. And if you go into the colliculus and look at what's going on in the colliculus, so here's the two colliculi. There's the midline. And we've got three layers to sort of consider. So we've got sort of superficial, intermediate, and deep. And the colliculus, remember the colliculus, the left colliculus deals with the right half of the world, so I'll draw it that way. So what happens? If we turn the light on, we find some activity somewhere in the superficial colliculus. And that's sort of sitting over a location in the deep colliculus that will make this saccade. So it will make a saccade that will basically move the animal's eyes to the light. And so that's what we talked about at the very beginning. That's the superficial retinal map over the saccade map. So this guy is deep, intermediate, and superficial. So we said that's how the map is set up. It's set up so that if you stimulate a superficial point, it will activate a deep point that generates the saccade vector that will bring the center of gaze to wherever the target is. So that's for a visual target. But now what if we do an auditory target? Well, an auditory target will actually generate some signal in the intermediate colliculus. So the superficial colliculus is strictly visual. The intermediate one starts to get other inputs, like auditory inputs and somatic touch inputs. The deep colliculus, that's the one that's actually doing the eye movement. And so that makes sense, because now this guy, that intermediate one, can go and stimulate the deep colliculus. And then that will bring whatever visual thing is at the place of the sound to the center of gaze. So everything's all lined up, everything's all good, animal's looking straight ahead. If you make a sound, it stimulates the intermediate colliculus in the same position as the light would stimulate the superficial colliculus, and those are both over the location in the deep colliculus that makes this saccade. So so that deep colliculus location here is the thing that's going up and making that saccade. Okay, that's all good, but what if the animal looks off to the side? So if the animal looks off to the side, so here's the same thing. So same animal, its head is fixed, hasn't changed its ears. So the ears are what is being used to calculate what part of the intermediate colliculus, using our favorite phase ambiguity resolution by comparing time between the left and the right ears, or comparing amplitude, sort of figuring out like where something is in auditory space. So it's all that stuff is happening before it gets to the superior colliculus. But now let's consider what if the animal sort of just looks off to this side. So its head is fixed, so its ears have not changed the interoral time delay. And then what if we just, turn on the same light or turn on the same sound? So now what's going to happen? Well, now we've got a problem, because if the animal's going to look at that sound, it needs to make a bigger saccade to look at that sound. And so what happens in the superior colliculus? What happens is kind of the inverse of what happens in VIP. So in VIP, what happened was we moved the visual input to match it up with the somatosensory input. We moved the visual receptive field here to match it up with the somatosensory one. What happens here is we move the auditory one to match it up with the visual one. So what will happen is, if you turn on a light, we're further in the periphery, because this is, the animal's looking over, if the center of gaze of the animal is over here, and so now it's further into the periphery of the animal. And so what's going to happen is the visual response is going to be, out further in the colliculus, into the periphery representation in the colliculus. So, and if we turned a light on, that you'd expect that to stimulate a portion of the deep colliculus that would make a bigger saccade. But now the problem is our head is fixed, and so if we didn't do anything, what's going to happen is the auditory is going to stimulate the same places it did over here, because the ears haven't changed. So what happens? Well, it just gets moved. So the auditory activation, if you have the animal look off to the side, so the experiment is just look straight ahead, put a sound on someplace, find out where it activates the colliculus, have the animal look off to the side, put a sound on, same sound on, see where it activates, and you'll see it activates a different place. And so, what happened? Well, eye position did this, right? Eye position did this. So eye position came back. It got recorded somewhere down here. It came back into the colliculus, into the, we left out a connection. So there's another connection there to the intermediate colliculus. So it came back in to the colliculus and moved the receptive field, the auditory receptive field, which shouldn't have moved because the ears were fixed. And so what does this imply? So this implies every time you're looking around, you've got the same thing going on with this auditory map. The auditory map is being shifted around the colliculus every time you look in order to sort of line it up with the visual map. And why is that? It's because what is the goal of the colliculus? The goal of the colliculus is to get the center of gaze on a target. The goal of VIP is to sort of like say, bite the target, do a somatosensory thing to a target. In this case, what the goal is, we want to get the center of gaze onto the target. In that case, we don't care where it is relative to the head. here we really care where it is relative to the head. Here we don't care where it is relative to the head. We really just care where it is, relative to the center of gaze. And that's what this procedure does, where the eye position information will come in and fix up the auditory map to line it back up with the somatosensory map. And this is happening like every time you move your eyes around, because you can effortlessly move your eyes around. A sound comes on, doesn't matter where your eyes are, you can look right at it. Okay, so there's just one final after dinner mint. So this is number C. This absolute, my final. So this is LIP pre-saccadic updating. And this doesn't just occur in LIP, it actually occurs in many areas. They've looked at it mainly in LIP. And so what is this? This is actually pretty straightforward thing. So we have 3 conditions. So here's 3 time points. So the first time point, the animal is fixating. Animal is fixating in the center. And the LIP receptive field, say the LIP receptive field is here. So this is so LIP has a visual map in it, so LIP receptive field is here. So how do we figure that out? We just have the animal fixate and pop little lights around very short while the animal keeps fixating and find out where the response is. So that's where the LIP receptive field is. Okay, so now what happens is the animal gets ready to make a saccade somewhere. So you need a target. Maybe you put a little target, a tiny target up, and turn it off. And so now the animal's getting ready to make a saccade. So what happens is, just before the saccade, so this part is fixate, and this is just before saccade, like 50 milliseconds before saccade. So just before saccade, what happens is, say the animal was planning to make a saccade to the right, just straight to the right. So what happens is, right before the saccade, the receptive field moves. So this movement occurs right before the saccade. RF moves right before saccade. So just before the saccade, then the animal makes the saccade. So at this point, the animal's still fixating, just before saccade. So that's what I mean, just before saccade. So still fixating. And then the animal makes the saccade, and then what happens, so here's that saccade. Say it makes the saccade to the right. It's the same distance as that. And so then what happens? Now the receptive field stays in the same place. So this is supposed to be same place. So how does this? And so now the animal's fixating. Now the animal's fixating here. So what does this look like? It just looks like. Let's imagine the receptive fields were stuck to the retina, and so every time you look around, there's no place to not get overlap the diagram. So if you look around, the receptive field just moves with wherever you're looking, like that. But now what happens here is I'm getting ready to make a move to the right. And so what happens is the receptive field goes, it preempts my move. It just says I'm going to move my receptive field to the place that's going to look at whatever you're looking at after you make the saccade. So then it just goes, this happens like every single time you make a saccade, and this superior click is getting up into your LIP every time you make a saccade. And so what does it do? It just goes, it moves the receptive field just in advance, then you make the saccade, and then it stays in the same place. So it just goes, and then like you're going to make a saccade up, it'll go, whoop, And so what does that do? Well, where does that come from? So this has to come, it's known that this has to come from, so the info comes from deep colliculus. This has actually been worked out because they showed that if you turn off this mediodorsal thalamus, that information from the deep colliculus about where it's intending to move doesn't get to VIP. It comes through the frontal eye fields. So the info comes from deep colliculus, via MD to frontal eye fields to LAP. So that's how it gets there. And what is the information? What is the information? It's, intended eye movement. So if we look at cells, in the deep colliculus, you find, some cells that only respond to the visual stimulus. So this is just visual. And then you find some cells that only fire before the saccade in the deep. So this is deep and superficial, superior colliculus. But then you find these other ones. So this one doesn't care about the saccade. It just says I'm just looking, there's a target there. This guy doesn't care about any targets. It just says, I'm just going to fire before the saccade. But there's other cells in the intermediate that do things like this. Maybe they've got a little visual response, and then they start building up. And what are they doing? They're sort of representing the intended eye movement. And then after they might even have a little spike at the end. So these cells were called build-up cells. But what are those guys doing? Those are the ones, and they're in the intermediate. Those are the ones that are basically programming the eye movement. The eye movement hasn't been released yet, because here's the actual saccade right here. So this is just information about what eye movement we're planning to do. And that information has to come up through this whole system from the intermediate and deep colliculus, go up to, MD to the frontal eye fields to get to LAP. And what does it do? It just essentially predicts what the world is going to look like after you make your saccade. And that's why the world doesn't jerk. Because what you do is, I'm getting ready to make a saccade to the right. Right before I make the saccade, the receptive field moves to actually look at what I'm going to see after the saccade. And then I make the saccade, and then the receptive field moves relative to the retina to sort of go back to where it was at the beginning. And so what it does is, essentially every time you make a saccade, it allows you to kind of see what the world is going to look like after the saccade. And then when you make the saccade, you go, it's all good. Nothing moved. And so this is happening. This whole system is all integrated, and this is happening after every saccade. And that's what sort of presumably gives you this feeling of, the world not jerking around, which it actually is. It's massively jerking around in all these visual areas. MT, VIP, LIP is jerking around all over the place, but you get the feeling of a stationary world because of essentially predicting what the world is going to look like using this information about this intended saccade that comes up through the system. Okay, so what do you think about that? So this was not reductionist. We kind of looked at the overall picture. This is everything's sort of going on at the same time. It's kind of hard to sort of keep it all in mind. And I kind of made up some of the stories, because nobody's recorded from all these things, 50 things at the same time. But they're just person, blind people looking at the elephant. We just looked at little bits of it here and there. But that gives you some idea of how it could actually work, and how integrated all different parts of the cortex are with the brainstem. So the brainstem isn't just sort of down and dirty stuff that you don't care about. It's actually it's right in there with, all the stuff going on in higher level cortical areas. Okay, any questions about, did I get it? Yeah, I unpacked it a little bit better this year than last year. Okay. So question, so this is the goal here is don't get your head bopped Over here, the goal is look at something, look at a sound, but taking into consideration where your eyes are, because your ears haven't moved. And this one was, let's take some of that information about what saccade we're planning to make and use it to shift the receptive field so that the receptive field can begin to see what it's going to see after the saccade. And then when you make the saccade, it just sees the same thing, and it's all good. It's amazing, like it's just happening like every 250 milliseconds, just shifting everything. All these different maps are all shifting around and sort of staying in register so that so you don't get confused and fall down. Yeah.
So the receptive field, is it kind of like a video game where it's like the target is like it moves this way, and then you look and then it's like fire, and fire is essentially you like fixating on that, and then it moves this way, and you look this way, and it's like bam.
Well, the thing about yes, good question. Like, what is a receptive field? Well, the receptive field in the retina, if you go to the LGN, that's nothing funny is going on in the LGN. It's just like, it's just telling you something. If you go to an LGN cell, its receptive field says something's coming from this part of the retina. So the receptive field corresponds to a part of the retina. So, if an LGN cell, responds to this part of the world, it's because if the retina is stationary, that's the part of the retina that connects up to the LGN. We're kind of extending the meaning of it here because in this case, the receptive field, like in the case of VIP, the receptive field can move relative to the retina because the animal has moved its retina. It moved its retina, like so. Here's its retina like this. It moved it up like that, and so that should have taken the receptive field with it. The receptive field should have gone up with the retina, but instead, some other information comes in and sort of moves the receptive field relative to the retina. So now it's in a more peripheral part of the retina. So the receptive field, the original idea of the receptive field was, what part of the retina is this cell hooked up to? But as you go up in the system, what you can see is that other things like the prepositus hypoglossy can go and move the receptive fields around relative to the retina if you need to. And the reason you need to in this case is because you want to know where a visual object is going to hit your head, but you don't care about where your eyes are. I mean, you have to compensate for where your eyes are. So if an object is coming in and you're looking off to the left, it's in your right visual field. If you happen to be looking off to the right, it might be in your left visual field. But you don't care about that. You want to know, like, where is it relative to my head? And for that purpose, we somehow have to move the receptive field around using some other information so that it's lined up with the face. I don't know if that helped. But I just say this is an extended idea of the receptive field. The original idea of a receptive field is, what part of the retina are you hooked up to? What are you hooked up to? The part of the retina that sees this, or the part of the retina that sees this. This sort of higher level version of the receptive field is, it's kind of like what part of the retina are you hooked up to, but what the eye position information has done is it's actually changed the part of the retina that is hooked up to the VIP neuron. It said, instead of being a receptive field that's relatively near the center of gaze, I'm going to change your receptive field, the place that causes the VIP cell to respond, to further in the periphery. So it's actually kind of like shifting it from where it's actually connected up to. That doesn't happen in the LGN. The LGN is just stuck to the retina. Wherever you're looking, the LGN receptive field will just follow it around. Okay, I put too much up here, but I just wanted to sort of give you an idea how the whole thing could sort of work together, and then you've got this integrated system involving like 50 or 100 parts. Yeah, you don't have to remember all this, and I never asked anybody about the prepositus hypoglossy E. Yeah, the equivalent of this, we can't leave without that. The equivalent of this in the vertical equivalent is called the interstitial nucleus of Cajal. Just had to write that up there. Interstitial nucleus of Cajal. That's the vertical vertical equivalent. It actually got a name. Okay, guys. We'll see you soon.